Good morning and welcome back, students. Today we are going to start with the topic tissue culture. That is a lecture 2 from Enhancement of Food Production. You have already studied about tissue culture in 9th and 10th standard. Tissue culture is a collection of different techniques. Here, isolated cells or tissues or organs are grown in vitro on a solid liquid nutrient medium. The part of the plant which is used in tissue culture is called explant. You know that plant tissue culture, it is based on the principle of totipotency. Totipotency is nothing but it is the ability of living plant cell to grow, to divide and to redivide and give rise to a whole plant. The method of producing thousands of plants in very short time through tissue culture is called micropropagation. So micropropagation is nothing but the technique in which variety of the plants are produced in very short duration. Micropropagation is also known as clonal propagation. It is the only process adopted by Indian plant biotechnologists in different industries, mainly for the commercial production of ornamental plants like orchids, then eucalyptus, etc., and the fruit plants like banana, grape, citrus, etc. The plants produced in the micropropagation process are genetically identical to original plants. So they are somaclonals or somaclones. Here is a chart which shows tissue culture technique. In the first step, cleaning of glassware, then sterilization of glassware and instruments in an oven or autoclave. Then the next, selection and preparation of nutrient medium with known concentration and proportion of different components. Then sterilization of medium in an autoclave for continuous 20 minutes under constant pressure of 15 inches. Then preparation of plant material or explant includes isolation of explant followed by surface sterilization and rinsing with water. Explant is obtained from the growing stock plant. Then inoculation of the explant in the culture flask containing sterilized nutrient medium. Inoculation is done in the laminar airflow cabinet unit. Now the plant which is inoculated, that is incubated, or you can say incubation of the inoculated explant. Here cells of explant grow, proliferate, that is multiply, to form callus within 2 to 3 weeks. Subculturing of the callus, that is, if callus is to be maintained for long period, callus is divided into 3 to 4 segments and then transferred to fresh culture medium. Then organogenesis, that is initiation of rooting and shooting, that eventually leads to plantlet formation. And finally hardening. Plantlets are transferred to polythene bags containing sterilized soil and kept at low light and high humid conditions for suitable period of time or for favorable period of time, and then it is transferred to field. So these are the steps already we have studied in the 10th standard. So just go through the textbook of 10th standard. In that biotechnology or introduction to biotechnology lesson is there. So in that lesson you can find all these steps. There are some advantages of micropropagation process. Micropropagation means what? It is also known as clonal propagation, in which many copies of plants or many varieties producing thousands of plants in very short time. Okay, so there are some advantages. It helps in rapid multiplication of plants. Then large number of plantlets are obtained within very short period of time. Then plants are obtained throughout the year under controlled condition. Then genetically similar plants are produced by this method. Therefore, desirable characters and desired sex of superior variety are kept constant for many generations. And the last, the rare plant and endangered species are multiplied by this method and such plants are saved. Advantages and the flow chart of tissue culture technique are given on page number 251. Let us now discuss about single cell protein. Single cell protein, it is an alternate sources of proteins for animal and human nutrition. For example, spirulina. You know that we get nowadays the tablets of spirulina for protein growth. It is a one type of blue-green alga. You can see here in the pictures about the formation from the blue-green alga spirulina, how the tablets and how the procedure is done to make it medicinal. Single cell protein, it is rich in protein, minerals, fats, carbohydrates and vitamins. It is grown on materials like wastewater from potato processing plants, straw molasses, animal manure and sewage. This also reduces environmental pollution. Advantages of single cell protein are given on page number 252. We just go through the points. Before we start with the topic animal husbandry, let us now discuss about biofortification. It is a method in which crops are bred for having higher level of vitamins, minerals and fats. Due to this, problems of malnutrition can be overcome. Following these are the objectives: protein content and quality, oil content and quality, vitamin content, micronutrient content and quality. So biofortification can be achieved through conventional selective breeding practices and also through rDNA or recombinant DNA technology. There are some examples of biofortification like fortified maize having twice the amount of amino acids. Wheat Atlas 66 has a high protein content and iron fortified rice has 5 times more iron are developed. Vegetable crops like carrot and spinach have more vitamin A and minerals. Vitamin C enriched beta gourd, tomato have been developed by IARI. So these are some examples of biofortification are given on page number 253 of biology textbook. Just go through, which is very important topic. Examples of biofortification may come for 2 marks. Let us now discuss about animal husbandry. It is a rearing of animals for commercial purposes is called animal husbandry. It is a agricultural practice of breeding and raising livestock. It is not only a skill of farmers but also is as much a science as it is an art. It deals with care and breeding of livestock like buffaloes, cows, pigs, horses, cattle, sheep, camels, goats, etc. It also includes poultry farming, fish farming, beekeeping, sericulture, lag culture, etc. Animals like honeybees, silkworms, prawns, crabs, birds, fishes, pigs, cattle, sheep and camels have been used by humans for the products like milk, eggs, meat, wool, honey, silk, etc. We'll discuss animal breeding topic later on in the offline lecture. Now we are going to start with dairy farm management. So it is a management of animals for increasing yield and quantity and quality of milk and its products. Milk yield depends on the quality of breeds in the farm. It is important to select good breeds having high yielding potential and resistance to diseases. In India, cows and buffaloes are mainly used for dairy farms. Sahiwal, Sindhi, Gir are Indian breeds. This is very important. Sahiwal, Sindhi, Gir are Indian breeds, and Jersey, Brown Swiss, and Holstein are exotic breeds, which are used in dairy farming. Buffaloes are restricted to some part of Asia only. In India, six breeds occur like Jafrabadi, Mehsana, Murrah, Nagpuri, Nili, Surti, which are all good milk producers. Cattle have to be well looked after. Quality and quantity of fodder in proper ratio should be given. Silage made from legumes and grasses, maize and jowar makes food feed. Silage is supplemented with oil cakes, minerals, vitamins and salts. Cleanliness and hygiene of the cattle and handlers is of more importance while milking, storage and transport of milk and milk products. The cattle shed must be cleaned daily. It should be spacious with adequate facilities for feeding, watering and lighting. Poultry farm management. Poultry includes number of bird species such as chicken, ducks, turkey and fowls, which are domesticated for their eggs and meat. So how poultry farm management is done? Components of poultry farm management: selection of disease-free and suitable breeds, proper and safe farm condition, proper feed and water, hygiene and health care. There are some poultry diseases like viral diseases like Rani Khet, bronchitis, avian influenza or bird flu. Then bacterial diseases like pulorum, cholera, typhoid, tuberculosis and chronic respiratory disease, enteritis, etc. There are some fungal diseases like aspergillosis and thrush, some parasitic diseases like lice infection, roundworm, sickle worm, etc. Protozoan disease like coccidiosis is a protozoan disease. Diseases of poultry are given on page 255. Let us now discuss about animal breeding. What is a breed? A breed is a group of organisms related by descent and similar general appearance, features, size, etc. Breeding is a modification of genotype of an organism to make that organism more useful to humans. For example, Jersey, it is a improved cattle breed. Then Leghorn, it is a improved chicken breed. Now what is the aim of animal breeding? To increase and to improve the yield and desirable qualities of animal. There are various types of breeding. The very first one is inbreeding. It is the mating of more closely related individuals within the same breed for 4 to 6 generations. So just remember that it is a mating of more closely related individuals within the same breed for 4 to 6 generations. By inbreeding, pure line of animals can be obtained. Inbreeding is helpful in the elimination of harmful recessive genes and for the accumulation of superior genes. Inbreeding has the demerits that it usually reduces the fertility and productivity. Outbreeding. It involves breeding of unrelated animals. The animals may be of the same breed but having no common ancestors for 4 to 6 generations. Now this outbreeding, it is having 3 types: outcrossing, crossbreeding and interspecific hybridization. Outcrossing, it involves breeding between the animals of different species. It is also known as interspecific hybridization. Outcrossing helps to remove the inbreeding depression. Crossbreeding. In this method, superior males of one breed are mated with superior females of another breed. So in which both male and female are superior, but they are from other breed. Desirable qualities of two different breeds are combined. The progeny hybrid animal may be used for commercial production or may be subjected to increasing and selection to develop new stable superior breed. For example, Hizardale and Bikaneri. Interspecific hybridization. It involves breeding of animals of two different but related species. It results in the formation of animals with desirable characters from both the parents. Such breeding is not always successful. For example, mule is a breed obtained from horse and donkey. Artificial insemination technique. It involves controlled breeding experiments. Semen from selected superior male is collected and preserved in the frozen state and then injected into the genital tract immediately in the female. It is useful to overcome problems of normal mating and convenience for transportation. So this is a simple technique in which semen from the male parent or semen from the male animal, it is directly injected into the uterus or into the genital tract of the female animal. So that is called as artificial insemination. The next is multiple ovulation embryo transfer. It involves a technology which provides the chances of successful production of hybrids. It is like a IVF technique which is done in the human or it is also called a test tube baby. In this method, cow is injected with follicle stimulating hormone to induce follicular maturation. Ovulation is brought about. In each cycle, 6 to 8 eggs are formed. The cow is either mated with an elite bull or artificially inseminated. The blastocysts at 8 to 32 cell stage are recovered non-surgically and transferred to surrogate mothers. This technology is successfully used in cattle, sheep, rabbits, buffaloes, etc. High milk yielding breed of female and high quality meat yielding bulls have been found to be successful to increase herd size in short period. So in this, cow is administered hormone like follicle stimulating hormone for follicular maturation and superovulation. So around 6 to eggs are formed at a time or per cycle, and then the animal is either mated with a bull or artificially inseminated, and whatever embryo is formed in the uterus of that female, that is removed surgically or without surgery, and then it is administered in the surrogate mother. So remaining part of the lesson we are going to study in the lecture number 3.